This video presents an introductory lecture on field measurements of water quality, including physical, chemical, and biological parameters. Of particular interest in this lecture are field sampling and laboratory analysis of these parameters. Recall some of the most common water quality parameters tested in the environmental sciences, such as temperature, dissolved oxygen or DO content, specific conductance, pH, turbidity, total phosphorus, different forms of nitrogen, alkalinity, hardness, and biological parameters such as bacteria counts, diatoms, and macroinvertebrates. There are several options available when sampling the above water quality parameters, and the application of various water sampling techniques will vary depending on the project's specifics, time, funds, personnel, and available resources. Parameters can be tested in the field using single or multi-parameter meters or test strips. Water samples may be collected and tested on-site using instruments, color wheels, or other equipment. Water samples may also be collected, preserved, and delivered to laboratory facilities for further analysis. Typical examples of parameters tested in the field with single or multi-parameter meters include temperature, DO, specific conductance, and pH. There are a variety of manufacturers, and each meter has specific requirements for calibration, that is, making sure the meter is set to known values of the test parameter, and verification, that is, making sure the equipment still reads known values after calibration and water testing. Procedures and documentation for instrument calibration and verification must be followed to validate any data gathered in the field. In general, these are quick to use and quite straightforward. Water samples may be collected either for on-site analysis or for delivery to a laboratory. A common sampling technique is the use of grab samples, which essentially means that the field team collects a sample directly from the surface water body. The field team may use the collection bottle, a long-handled dipper, or some other tool, but whatever is used must be properly cleaned and rinsed prior to collection to avoid cross-contamination. On-site testing may include a turbidity tube or portable turbidimeter, use of colorimetric testing field kits for nutrients or other parameters, or pH test strips as examples. In many cases, a minimum of 3 replicates are desired, as 3 is the absolute minimum number of samples needed to report a mean value with associated variance. In some cases, water samples may be collected for delivery to a laboratory for more sophisticated testing. The sample is generally preserved on ice, though other preservatives may also be used depending on the parameter of interest. For example, nutrients may be fixed with an acid solution to prevent changes between forms. As a general rule, most parameters should be measured within 24 to 48 hours, with few exceptions. Common parameters covered in laboratory analysis include several forms of nitrogen, such as ammonia, organic, nitrite, nitrate, and total Kaldal nitrogen, which is a combination of ammonia, organic, and reduced nitrogen. Phosphorus, generally as phosphate or total phosphorus, alkalinity, hardness, and turbidity. Generally, laboratory testing requires meticulous cleaning of glassware, expensive technical equipment, and great expense, but the readings have a higher level of confidence and precision than quick field test kits. Laboratory personnel are trained in one or several instruments and are responsible for running blanks, standard concentrations for verification, routine machine maintenance, troubleshooting, and upkeep. Those of you with an interest in chemistry may seek a laboratory position as a water science chemist. Laboratory analysis is typically used for bacteria counts and metals as well. This slide presents only a handful of possible methods for detecting such contaminants in the water column. Such techniques require specific growth mediums and reagents, but are fairly simple procedures, though you will need access to an autoclave to sterilize glassware and materials, and in some cases, access to a sterile hood for plating. The references listed below were consulted in developing this lecture.